Hello, everyone. My name is Anjani, and I'm a conference director with Cambridge Innovation Institute. I produce several of the tracks at our Discovery on Target event that's coming up in September in Boston. And I'm delighted to have today one of the speakers with us, Dr. Stephen Fesik. He's a professor at Vanderbilt University, and he'll be speaking on my Emerging Drug Targets track about his work on MYC. So he's going to spend a few minutes to preview his talk and tell us more about what to expect and his work. So, Stephen, well, first of all, let me give you a little background on c-MYC. MYC is a transcription factor, and it plays a role when it's overexpressed or mutated in many cancers. And it's been a very elusive drug target. There's still nothing on the market for it. In fact, when I was in grad school over 30 years ago, one of my first lab rotations was working on MYC, and it's still quite intractable. So Dr. Fesik has worked on many cancer targets that are hard to drug, including KRAS and BCL2, which led to a lot of progress using fragment-based approaches. So, Steve, one difference, though, between those other cancer targets, difficult to drug cancer targets like BCL2 and KRAS, which he has presented before for us at our discovery events. In fact, you were a plenary keynote at DOT a few years ago. So you targeted the protein-protein interaction sites. So the sites on BCL2 and KRAS that interacted with their partner, and that's how you disrupted its function. I know MYC is a transcription factor, as I said, so it binds DNA. And so I'm wondering, is there any difference in your approach in targeting MYC over your previous successful cancer targeting projects?
Well, there's two ways that we've targeted MYC. One of them is blocking a protein-protein interaction. It's blocking the interaction between MYC and its cofactor WDR5. So that is more analogous to other things that we've done before, and that's far along in our studies. The other way, which you point out, is to go after MYC. When complex with MAX, it acts as a transcription factor, and we're trying to block the interaction between MYC, MAX, and DNA. Now, this is thought to be extremely difficult. And frankly, we didn't know when we started the program whether we were going to have any success. Nonetheless, we prepared the MYC-Max dimer and did a fragment screen. And luckily, we found molecules that bind right at the interface where the DNA binds to the protein MYC-Max complex. And the difficulties we're having now is optimizing the molecules. Because as you might guess, when you want to bind to a protein that interacts with DNA, it's going to be a charged molecule. And it's not going to be, although we can get the potency, it's hard to make drug-like molecules. But we've succeeded using various tricks, and we have molecules that potently bind to the MYC-Max dimer and block the interaction with EBOX DNA.
So what stage is this work at now? Is it in preclinical or
The
further?
work on WDR5 is actually ready for IND enabling studies. We have a very potent molecule that binds to the Win site of WDR5, and this molecule is ready for IND enabling. We've worked out the formulation, the chemistry, all the things we needed to do, and now we're ready to test it and then eventually test it in the clinic. For the MYC-Max dimer, we're still optimizing the molecule. We think we need more potency, and that is what we're trying to achieve by various means, but it's moving along. We've made rapid progress in improving the affinity of our compounds over the fragment hits that we found originally. And these molecules that we found potently inhibit the binding in EMSA assays.
Now, how long you've tried MYC before going after it? Is this a new strategy or anything new recently
Yes.
that you've done?
We wanted to go after RAS and MYC. These are the two big players in cancer. They're highly validated targets. So we chose to go after them. RAS, we had success as we presented before, but MYC was lousy. That is, we started with going after MYC by itself, going after the DNA binding domain of MYC and going after the transactivation domain. We cloned and expressed both pieces of MYC and saw that they were unstructured in solution. And you can tell that from the NMR spectrum.
You say binding domain, you mean DNA binding domain? Or,
Yes,
okay.
DNA binding domain. Now, this is at the time at which unstructured proteins were really popular in the literature, and everybody wanted to go after these unstructured proteins. It didn't make sense to me that you could get something that would bind selectively to an unstructured protein.
That also intrinsically disordered, is that
Intrinsically,
another term for it? Okay.
yes. And so, but nonetheless, we said we've had odd things come up before. Let's do the experiment. And both domains did not bind anything in our small molecule library, which shouldn't come as a surprise. The pockets we usually tackle are defined pockets, and we find molecules that bind to these. The intrinsically disordered proteins, they didn't bind anything. So we dropped the project, and that's our start in the MYC field. But fortunately, although we couldn't target MYC alone, the MYC-Max dimer does indeed adopt the structure, and we do find hits in our fragment-based screen.
Okay. So it's not a intrinsically disordered
No.
structure, this new area you're going after. Okay, great.
Yes.
So just now to go a little broader in terms of your approach, you always use fragment-based approaches, I understand. And I mean, you are kind of a pioneer in the field. I know over 30 years ago, you had success at AbbVie. I think that's where it all started. You published a paper on SAR for NMR, something like that. But so what has made you stick with fragment approaches, or why have you had success with them?
So, in my view, fragment-based approaches, structure-based design is the way to go. We have had amazing success going after so-called undruggable proteins, regular proteins, and we like this approach very much. But the key, I think, to our success is the methods we use. I was, quite frankly, kind of shocked that when I took part in the medicinal chemistry series, where we reviewed the publications in the last year, and this has been going on for 10 years. So I looked at all those that publication for 10 years, and I saw that not many people use the methodology we use. I believe that the way to go with fragment-based approaches is use NMR as a primary screen and use protein observed NMR on isotopically labeled proteins. If you have a small protein, you can uniformly N15 label it and run a standard N15 proton correlation map. As you go larger and larger, that's not a limitation these days. You can screen proteins. We've screened, let's say, 66 kilodalton proteins and higher. And the trick is you isotopically label with C13 the methyl groups of certain amino acids, and you can clearly see signals. They're narrow enough, and you can follow the shifts, and that's how you can do your fragment-based screen. But this method, by looking at the protein, is doesn't have many artifacts and is the way to go, we think. And the other trick or the other feature that we apply is our library is carefully formed, that we have a library of about 14,000 compounds. Most people use libraries that are much smaller. If I had my choice, I'd even go larger, but it's not that practical to use Protein Observe NMR with a larger library, at least not for us. So I think these led to our success over the years with fragment-based approaches.
So you use the same library for all your projects, or?
Yes, we
Okay.
used that library. It took us a long time to prepare, to get out the bad actors, so to speak. But now I think we have a really good library for fragment-based screening.
Hmm. So many others, they would, rather than labeling the target, they're labeling for NMR, they're labeling the small molecule.
You can either do not label, and you can follow the proton signals of the ligand, or you can label with fluorine 19. There are groups that are doing this. It's faster, it's easier to prepare the proteins, easier to collect the data. However, in our experience, the protein observe method that we use is the gold standard.
Okay. Well, this isn't a fragment-based meeting, although we do have those, but I think this might get into a little bit of the weeds for non-fragment people, but Dr. Fesik will be there for the fragment people to talk to you more one-on-one. Also, in San Diego, we have a little bit more of a fragments focus. We also have our lead generation strategies track that will be running in parallel, and people can hop around from track to track at the event, and there will be more fragment coverage there. So, whether you're interested more in the cancer aspect or medicinal chemistry, or your hardcore fragment-based drug discovery, I think there'll be something you'll be covering, all of that in your talk. So we look forward to it, and thank you so much for spending the time today. And I'll see you in 2 months in Boston. So everyone, if you want to see more, see Dr. Fesik's talk, it's on the Emerging Drug Targets track. Go to discoveryontarget.com. You'll see all the agendas there. You can also search for speakers, and then it will take you to where they're speaking at the event. So thank you again, Stephen, and thank you, everyone. Goodbye.
Thank you.